Good morning, viewer. Welcome to our FTV English version. Viewers, picture a living cell as if it were a city. Yeah. If you were the urban planner for this very alive city, one of the things you would have to decide is how to allocate space for different functional uses. Parts of the city should be zoned for residential use, for example, while others should be zoned industrial. At the same time, you have to put the trash somewhere. Ideally, all of these essential activities should be compartmentalized so they don't get in the way of the others. As a city grows, its different compartments also need to grow. This is due to in a living cell too. But do all of the pieces grow at the same rate? How do you assign the resources to grow? Up until now, scientists have had very little visibility into how the cell organizes and prioritizes the growth of its compartments. The functional structures known as organelles. This includes some of the more familiar organelles like the cell's nucleus or its energy making factories called mitochondria. In a new study led by Shankar Mukherjee, an assistant professor of physics in arts and science in Washington University in St. Louis, is one of the first to take a system approach to measuring and interpreting the changes that occur in organelles as living cells grow. His team conducted their research published 6 June in Cell Systems using rainbow yeast cells as their study system. Yeah, we used a technology known as hyper spectral imaging, which lets us label virtually all of the major organelles in the cell so that we can directly observe how the cell allocates space to these compartments, Mukherjee said. Although his team did not invent hyperspectral imaging, Mukherjee said they are among the first handful of researchers to apply to understanding cell growth. The new approach allowed them to simultaneously visualize 6 major metabolically active organelles rather than looking at a single or pairwise correlations among the organelles. As had been done in the past, the physicists could set up their experiments and then let the data tell them what was happening with all of the organelles at once. Amazing, sir. Behind this exclusive report.